in the Michigan State. A lot of places now hire new faculties, have a specific focus on the quantitative genetics. Purdue, Washington State. So it's not the old quantitative genetics we're talking about. It's the new era quantitative genetics we're talking about. And I also want to give compliment to Dr. Jim Spetch. And I think my first quote unquote invited talk was given at the ASC meeting. At that time, Dr. Spetch and then Perry Cragen was organizing a genotyping workshop. I was a postdoc at Cornell, so I volunteered and then invited to give the first talk at the ASA, which was a great meeting. I went back to check. That was the first presentation I gave. Thank you very much for that. Now today my talk is right after his. Yeah. Here I don't want to try to brag out my research, but this is a plant breeding symposium. There's a grad student training component in this. So I want to show that when I was a grad student, what I did, that was back in 98 to 2000. I did a master with Mitch Tynstra, who's now a maize breeder in Belgium at Purdue University. At that time, he was in Kansas State doing the drought tolerance. This is the thing I published out of my master research, this genetic analysis of the drought tolerance. But then the title, did not actually reveal that at that time, I was learning the basic design too. You have a set of male plus a set of females, the genetic design there. I was calculating the general combinability, specific combinability, and I did not realize that was very relevant of what I learned here later on in my career. And in this talk, we just did a greenhouse, goose chamber experiment, try to correlate what is observed, some like in theory, with the field performance in practice. And then later on, I did my Ph.D. with Rex Bernardo, did some small project. Here, this is where I learned about the Steve Stock synthetic and then non-Steve Stock, Ohio 43-17, that heterotic pattern. And, this very small publication gave me the opportunity to learn some of the things that are concrete, that are beneficial to my rest of my career. And then this other project was doing about genetic variance components estimate. Advanced cycle breeding, you estimate the genetic variance component from the studies, so that we say whether genetic variance is decreasing or increasing. And here we did something from metabolic control analysis. So metabolic control pathway is the theory behind epistasis. So that's something I learned about epistasis. And later on, we did a one-year postdoc with Rex Bernardo. Here we learn about, this essentially is something called GWAS, but that time we don't have enough markers. We're just setting up a model. In this publication that set the stage is a mixed model, QK mapping, and that time we were actually doing more complicated models because of the hybrid performance as a phenotype. And then later on, I sort of gained my nickname. I went to a meeting and some students will say, oh, you're the E et al. So this is my nickname. So it's because of because people are more familiar with probably two or three, two of these publications. One was the Nature Genetics publication where we did a QK mixed model. So you could talk about this forever, but really what we did is reintroduce this mixed model framework under the context of genomics. It just really, people liked it. So really actually influenced a lot of study, but also make it difficult. So it used to be we're just mapping easier, but after we introduced the mixed model, things get more complicated. You have to Q and K and then later on we did this nested association mapping design. I think it happened that Jim showed that nested associated mapping, the diagram. So we did that. That's published in Genetics. And later on we did also, after I started at K-State, we did a simulation study about genomic selection or genome selection for quantitative traits with Rex. This turned out to be one of the earlier papers that in genomic selection that is well received by the community. And then my postdoc and student that did all other quantitative related studies. And then we have other studies I'll briefly introduce later on. So what I'm saying that as grad students or postdocs in plant breeding, agronomy, this general area, you learn the basics from a small project you were assigned, but if you actually expand your knowledge while doing the research, I did not stop at design 2. I learned about design 1. I learned the diallele, and then I learned all this together. And then I found it very interesting, very helpful that now we're using this knowledge to apply to the genomics data set. So, and then over the years, I always try to think of what our role. I mean, as a scientist, always thinking, what are my potential contribution areas? So 2 questions I like to ask is that, what is the most efficient ways to identify gene underlying complex traits? This actually is very challenging because we know that complex trait is controlled by multiple QTLs, their interaction, and also an interaction with the environment. I mean, it's so difficult to play with this. There's a lot of things has to be involved, right? And then another one would be, how can we incorporate genomics and phenomics technology to improve the current breeding process? If you listen to some of the presentations by Taveri and other speakers from industry, what they're saying is that the plant breeding is constantly evolving and also exceedingly successful. So that's the thing you want to improve. So, and it's very difficult because something has been very successful, you want to improve it. And then, so that involves a lot of genetic, gain equations, breeding cycle, reduce that, and then or speed up the breeding cycle, germplasm, trait complexity, and logistic support. And then years ago, I wrote a vision paper or perspective paper in the plant genome. At that time, we always talk about what is the potential for the ultra high throughput genotyping. I mean, we're thinking that's going to change the world, but actually the fact it's not. So besides the genomic technology, we also need that methodology to figure out the G to P relationship, genotype-phenotype relationship, mutational screening, progression library, all this research has to be carried out. And also the tools about double hyperload and transformation, screening, and phenotyping, all these are all very important because of the sequencing and genotyping was mainly driven by medical research. They actually really push that forward. All the other things that plant breeders and geneticists, we have to do our own because, plant, we were actually grew out in the field, right? They're not like humans. They'll line up in the doctor's office to get their phenotype, right? We have to go to the field, phenotype. It's very challenging. And then breeding methodology, and also I think Taveri focused on these people. So the training of the next generation plant breeders is getting challenging because when I was a student, I did not have to run any jail, and I did not have to do any, like a GBS data analysis. I mean, look at all these young grad students here. I think you guys do a great job and better than me because I didn't have to worry about all this high throughput thing. It's a lot of work. And then this position, actually, I just recent hire at Iowa State. I put this position description here as that kind of an inspiration for me, incentive for me, because this was well written. This was a sentence that attracted me to apply for this position and later get offered, is to combine maize breeding with cutting-edge genomic technology to address significant questions in quantitative genetics and develop and improve contemporary breeding methods. Everybody knows that Iowa State is a place that with this legendary names, so it's a tremendous amount of pressure for me to do something that are useful to the community. So I go back to review some of the textbooks in quantitative genetics. On the left side, there are just 2 legendary books. They're very comprehensive. It's amazing that this title of the book called Introduction to Quantitative Genetics. If you can just be good on a few chapters of this introduction, you're good for your career, actually. It's very, if you look at Farkas' book, I think Lauren was actually using this book, right, Lauren? Teaching some of his quantitative genetics class. I mean, it's very dense. And also, this some of the people call this new Bible, this new book is, Lynch and Voice, also very extensive. There's some mathematical part. You want to get into the detail. This too is actually, this used to be the yellow cover page by Anna Haller, and it was updated where it became a green, kind of different color. And this was by Rex Bernardo, was my Ph.D. advisor. There's more breeding components here. They're just theoretical ones. But I think I picked some of the goals of quantitative genetics. This hasn't been changed. Is the nature of quantitative trait variation, consequences of inbreeding and outcrossing, the constraint of the evolution process, estimation of breeding values, getting more interesting here, and the development of predictive models. If you look at the last two, they're about breeding, prediction, selections. The first, maybe the first one or two, the first one is the nature of quantitative trait variation about gene finding, GWAS, QTL stuff. So this is the general framework of quantitative genetics. So in the recent years, we have been, doing some of the research. This too, I'll just show one or two slides about multiple domesticated alleles and their distribution, shattering genes in sorghum and also other species. And then this was a recent work we did about genome-wide genetic polymorphisms underlying quantitative trait variation. So this is actually a study we tried also bring back to the quantitative genetics framework. And I'll show, a few studies and just, quickly flip through them to give a taste about the combination of quantitative genetics with the genomic information. How do we do things better? And then these are the things that we try to do. What we just started about crop modeling and epicene in genotype by environment interaction. Long-term project again is very challenging. Analysis of framework data from the high-throughput phenotyping. I mean, we've been really trying to optimize this phenotyping scheme and then all this great stuff. But also I think we start thinking about what is the analysis framework where we're going to analyze this data together, and then about heterosis too. So this is the first study we did about the parallel domestication of shattering gene in cereal. After this was published, I got a lot of questions because people don't understand why a quantitative geneticist want to do this, QPL cloning project. And this goes back to Dr. Spetch, the quote, something in theory, everything is good. Quantitative genetics, we talk about big A, small a, and 2PQ alpha square, all those mating designs. But and then when I studied this by position at K-State, I want to see exactly what is this big A, what is a small a, what is the wild type allele, what is a mutant allele. So we started the QTL cloning process, try to figure out the genes, exact genes, not only mapping the QTL, fine map the regions, but exactly gene control the shattering of the habit of this wild type sorghum. This is actually domesticated sorghum. So what the gene cloning stuff always interesting, it got published, but what the quantitative genetic side of this story is, if you look at upper here, this is the wild type allele. So we actually shaded with same color. We have 25 examples that have excisions have this wild type allele. And then, but if you look at the domesticated allele, there's 3 different types. There's this GG type here. There's this G2A, A to G, and this type, we have 37 excisions, and then 47 excisions. The story that in original biparental mapping populations, we had TX430 and the sorghum vagatum. And then you clone this gene, you find this possible gene, and then you have the haplotype of this domesticated allele and the wild type allele. But then you sequence another wider excisions, you find these exceptions. This one would actually, before you figure out this one, you would say, can I get a different pointer actually? I have a pointer in my bag. Sorry about that. So before you figure out this mutation, which is a splicing type mutation, this was actually wide counter argument to say this is a functional site because they're similar to the wild type. And then before you figure out this one, also this one is a large chunk of deletions. So in summary, there's multiple domesticated alleles unless you figure that out. So instead of big A, small a, wild type and mutation, we're talking about big A and small a-1, small a-2, small a-3. So this study was also, after we figured out the sorghum gene, we figured out 2 copies of maize gene that is potentially be controlling the teosensate to the maize and also a rice gene. So that was a parallel domestication in the shattering cereal. Here, there's another recent study we published in Genome Research, and then about genic and non-genic contributions to natural variation of quantitative traits in maize. This figure, I was not trying to let you see those different figures, but basically what it says that sometime they do a GWAS mapping, the signal is landed on the genic region. Like we like to see a stop codon, we like to see a major change, a deletion, within the intron, the exon, like splicing site mutation, but sometimes you'll land it in the upper, the promoter regions. So the signal can be on the genic regions and in non-genic regions. Here, basically we're saying that using some of my quantitative genetic skills, I understand using the NAM design, nested association NAM design, because you select 25 founders, making a cross, and then populations. So the chance to be detected of your QTL is not only depending on the genetic effect, which is the case if you do a parental mapping population, but in nested association mapping panel and also the GWAS, it's not only depending on the genetic effect, but also depending on the allele frequency. So now I go back to the 2PQ alpha square additive variance. So for the genetic effect to be large, you have a unfavorable allele frequency. It's okay, it can still be detected. That's a shape going up. So the chance to be detected is a bar on this plot. You either have a good frequency, small effect, or large effect, have a less favorable frequency. It can be detected. So now move to some of the examples about how we apply this quantitative genetic principles into the more genomics or gene finding research. The first one is a multiple recessive allele over non-tanning phenotype. And then when we started this project, we went back to the earlier study from classical genetic analysis, indicating there's two loci, B1, B2, control the tanning presence, absence in the sorghum germplasm. Why we start tanning, I'll introduce in the next slides. And then we have a linkage mapping, identify two QTL, one on chromosome 4, one on chromosome 2. So that was just easy, and it was straightforward. So, and then we published this TAN1 paper, and then a few years ago. So the reason why we start tanning is that tanning is somewhat interesting because it has a disadvantage of astringent flavor. It has nutritional molecular precipitation. A lot of sorghum, 99% of sorghum grown in US is actually non-tanning sorghum because the feed efficiency will decrease if you feed non-tanning, feed the tanning sorghum. But tanning has its advantage. It has a defense mechanism, antioxidant, and also be cold tolerance. Interestingly, that farmers in Africa, some areas, farmers would eat tanning sorghum, right? Because tanning sorghum would actually make you feel more fullness, so you're not hungry when you go to the field. So that's actually a little bit sad. So now we're talking about maybe we should actually feed a lot of developed world with tanning sorghum so that you don't eat much, you don't get too much energy, right? And then, so tanning is actually the testa layers under the pericarp. And also tanning sorghum, actually interestingly, if you actually in Africa, the birds, if you have a tanning sorghum and non-tanning sorghum, or pick on the non-tanning sorghum, they'll leave the tanning sorghum alone. So it's like bird resistance. So when we were publishing that earlier study, we figured out there's again there's TAN1, which is wild type allele. There's TAN1-A, TAN1-B, that's the mutant allele, give you the non-tanning. But after publication, we figured there's another one called TAN1-C allele. So all these alleles is actually the TAN1 gene actually have the WD40 domain. This is WD40 domain, 4 of them. This is the wild type allele protein sequence. If you have the mutant, it actually get problems. And then TAN1C also give you a problem because actually extended the amino acid sequence to be much longer. So again, now we are because of now we're talking about the high throughput genotyping. Earlier study, we used just regular, SS markers, and then meta-QPL analysis, and then the sequence of candidate genes. Here, what we did is actually using a different population, and then we use the GBS SNPs, genotyping by sequencing, about 20K SNPs, and then we were able to remap this the second gene, and then again narrow down to 89 KB region. And then if you look at the candidate gene underlying that within that region, and then there's one gene that stands out because the tannin biosynthesis pathways have been very well studied in Arabidopsis. So another story here is that when I was a grad student, postdoc, I never liked the Arabidopsis work because we couldn't use it. But now after the sequence of the sorghum genome and all this other crop species, I think that actually now is the time we actually can enjoy the benefit of this model species because we actually use that exactly in our earlier publication of this TAN1 gene, and then now we're using that again for the TAN2 gene. So again, this TAN2 gene is actually a BHLH transcription factor, and then again we find three mutant alleles. That's a wild type TAN1, and here TAN1, TAN2A give you there's a 5 base pair insertion here, another insertion here. There's 95 base pair deletion here. I'll talk about this frequency of 3 different alleles. As you can see that a matching process is not always easy. You get single base pair deletion, everything the same. It actually have diversities here. Now what we did is similar to earlier study, we have wild type Arabidopsis. Arabidopsis is a weed, so actually it has a tannin in the seeds. It's a brown color, which is revealed by the brown color. And then there's actually this is actually a TT2, which is a homologue of the TAN2 gene. And mutants, I give you this non-brown color, which has non-tannin. And then if you actually do a transgenic experiment to put this TAN2 gene, the cDNA of the TAN2, into this TTA mutant, you can recover this tannin. Also, we can see that there's, induce a little bit of flowering time or the elongation of the stem. But then we were able to do this easily because, doing transformation in sorghum, it's possible, but it takes longer time. This actually is easy. So here's actually the most interesting part of this quantitative genetics in the gene cloning process. We know that these two genes is a complementary dominance. Complementary dominance is actually, we are talking about under our F2 conditions. You have to have one dominant allele of each of the two genes to give you this tannin presence. Within the homozygous state, so the real population, you can say that you have to have big TAN1, big TAN2, to, homozygous to give the tannin, which is the solid circle here. And then if one of the gene is recessive or mutated, you get non-tannin. So you get this full, 2x2 tables of 92. These numbers are the number of oxidations that we studied, number of sorghum oxidations we studied. So this actually would you could think about this big A, small a, big B, small b. That's regular we talk about in genetic class. But then what is more interesting that if you expand this small TAN2 with a small e allele, you can actually find a -A, -B, -C allele, -A, -B, -C alleles. Now instead of a 2x2 table, you get a 4x4 table. And then this cell is the same as 92. But then these other 3 cells will split into another, let's say, 15 cells. And you can see that what is more interesting is that we always talking about GWAS and mapping. And what is interesting that TAN1 gene, it has a favorable frequency, so that you have about 57% of the TAN1 here, and then TAN1-A have 24, and then 12, and then 5. If you're doing the GWAS mapping, it happened that it's great that this allele has a higher frequency that allow you to map this gene, because these other alleles will provide a counter argument to this gene, this mutation. This too will be lumped into this tannin gene, which will not align up with your phenotype. But it was interesting that for TAN2, you see the frequency is 3%, less than 1%, and then 10%. So if you actually do a GWAS study, even if I tell you this is the gene, you will not get a peak. That's a lot of questions we got asked. Why we did all this money, we spent this phenotype. Our phenotype was robust, was indicated by the repeatability. Why we don't see the peak on our interest candidate gene? So that's one particular reason. This trait is very simple, and there's only 2 genes functioning, and then you're supposed to see a peak. But if you did not see a peak, it might be because of frequency issues. There's multiple causal mutations, and then the most frequent mutations still not frequent enough for you to be detected. So that's actually what we say. GWAS was not able to detect the TAN2 gene just because of split of the frequencies, unfavorable frequencies, and we need an integrated approach in trait dissection. We need actually conducting this QTL cloning, mapping, and then function prediction together with GWAS, combine them, and then you'll be able to validate this gene underlying the trait. This is a study we've done. We're still ongoing. It's about the missense heritability and the rare variants. So many of you familiar with the missense heritability term. This is after all this great GWAS study, and then this is actually done in human medical genetics. After you summarize the specific genes that has been validated after its initial GWAS discovery, the amount of variation explained is actually far less than the estimated heritability. So the estimated heritability is the upper boundary of the contribution from genetic components. But after we clone those genes validated, there are actually sum up to the very small proportion. That's a missense heritability. So the reason, again, now go back to theory. In theory, there's all these possible reasons: structural variations, PEVs, and then epistasis, G by E interaction, heritable epigenetic factors of rare variants that can explain the missed heritability. This is a very hot topic, and sometimes we call dark matters in the universe. We can feel it, we can estimate it, but we cannot finding these genes and to sum up, to make up to the proportion that we want to explain. We have a postdoc used to work in my lab, and later on he moved to a medical school. It was actually sad for us to lose him. He did a theoretical contribution, which is basically saying that if I can assume a distribution of genetic effect size, which is a double exponential distribution, and allele frequency classified as rare variants, which is the Mali allele frequency under less than 1%, low frequency as 1% to 5%, and then the common variant is greater than 5%. This is the definition from the human genetic area. And then also you want to define a relationship between genetic effect size and allele frequency, which this is actually the tricky part, because a lot of things we don't know, but we have to simulate something. So here, you can see that rare variants, we simulate it to have relatively higher chance to have larger effect than the common variants, which is the red line here, that small effect. If you take that, and then we did a lot of simulations, we think that rare variants only contribute to a small proportion to the overall genetic variance. Then again, that goes back to the basic quantitative and population genetics is a 2PQ alpha square. Alpha square measures the additive, the average effect of allele substitution in square, but the P and Q are the frequencies. If the rare frequency by itself, if you have 0.1 times 0.9, it's only less than 1%, right? But if it's 0.5 and 0.5, there's 25%. So the frequency issues come into play here. And I'm working with some of the human geneticists on the CHARGE consortium data is the cohorts for heart aging research in genetic epidemiology. We were able to get access to 962 European Americans, and with 25 million SNPs on the 22 autosomes. Now this is actually the big data set that we have to deal with. And then we were able to do 6, sort of quantitative traits. We are now looking on more of this trait, more of this, more traits than the 6. Body mass index, height. The height, human height, this is the trait that highlighted with the missing heritability issue in the, the popular journals or the scientific journals. And then the LDL, HDL, and triglyceride, and also total cholesterol. So here is actually exactly what we did. Again, this is a kind of a theoretical estimation based on empirical data. We always try to combine the theory and practice here. We used a mixed model. This is a trait, this is a fixed effect, and then this is a random effect. So if you remember, if you're familiar with the QK mixed model, so when you construct a kinship model, you were using the genome-wise SNPs to construct a kinship model. Okay, we use the same strategy, but here we would actually divide our SNPs to different allele frequency bins, and then using only SNPs within that frequency bin to calculate the relative kinship. Then we would have multiple kinships. Instead of having one kinship, we'll have multiple kinships. And then they'll have, instead of one random parameters, we'll have multiple parameters. And then we'll have an overall estimate of each parameters. So here is what we did on those three traits, BMI, height, and then LDL, HDL, TG, and TC. Again, there's you don't have to see this exactly, small numbers. It's just a pattern. Let's use this example. On the x-axis is contributions to phenotypic variance. And then the first black bar is if you use all the SNPs all together to calculate the kinship matrix. That is your genetic variance estimate over the phenotypic variance, about close to 40%. If you only use the common SNPs to estimate, you get less. But if you actually put common variance in one genetic principal component and then low frequency and rare frequencies, you are getting closer to the black bar estimate. And then ignore this part. This will just further dividing the common frequencies in different bins. It's not that relevant. The most important bar is that this second bar here. So you could see that in many cases that you could say that this tip that colored with blue is the potential maximum contribution of the rare variance to the overall phenotypic variance. And then now it's depending on whether you are optimistic, you are pessimistic. Okay, so we say contribution to rare variance to the total phenotypic variance range from 2 to 8% for 6 human quantitative traits. If you're optimistic, you say, great, I'd be able to count for this many. But if you're pessimistic, say, oh, that's not much at all. But and then we say empirical detection and the validation of a rare and common variance would further enrich our understanding of the relationship between the allele frequency and genetic effect size. In this calculation, we did not have to validate each causal variant regardless of frequency. We just have an overall estimate. This is what we get here. And then we mount to the third example of the G by E, and it's not a typical G by E would assume, but then we try to map QTLs and networks. And so we were actually doing the sorghum NAM populations, and one of the populations we discovered is actually from TX430, this the green sorghum line, and then P898012. This was released by Purdue, Gabisa's program. And then it's interesting to see this PR is Puerto Rico. And then we observed that in Puerto Rico, this line flowered early. See, this is not late. But in Kansas, if you drew the same line here, this actually flowered. This is actually still going on. We have to wait until class started, and then I still have to go to the field while I'm teaching. Undergrad helper is gone. It's a mess, right? But you talk about the faculty's life, and then we have this exactly switch. Right, crossover interaction of the phenotype on those two parents. We say, well, let's put some students and then see what we can find out. And here again, this Puerto Rico is close to the equator area. It also corresponds to the area where the sorghum was originated as a tropical C4 species. It's a photopure sensitive, right? And then this is Kansas, and then this Iowa. We moved to Iowa. And then if you look at here, this Puerto Rico, Kansas, Iowa. And then this blue line and the red line, this P898012, I mean, this is like textbook style of the crossing over interaction on these two parents, right? Here is early and it's really late. Look at here. And then here's stable. And then, Iowa, what happened in Iowa is shrinked. I would imagine it keep going up. So there's a lot of this temperature and photoperiod combination interactions here. And then this is the reals, recombinant red line, reals. You see that some of the real will follow patterns of one parents, and then the other parents, and then again, there will be other recombinants. They have a different pattern, which is buried in between. So they have, and the overall pattern you see was perfectly, mimic whatever we observed in the parents. And then there's nothing fancy here. What we did is actually we did a GBS, and then on this population, and then we detect 3 QTLs using the phenotype from the Puerto Rico, and the chromosome 6 and 9, and then the chromosome 6 and 8 and 10. And then if you do the same thing here, okay, upper panel was done by the single mark analysis with the GBS SNPs, the physical distance on the chromosome length. But again, I mean, a lot of people will get trapped into all this, but actually what we did actually, we should always go back to the genetics, because the genetics are fundamental behind this sequence data. If you are now going back to the centimorgan genetic distance, you can see this wide span of the chromosome 6 only occupy a small fraction here because there's less recombination in that region. All this up and downs is because of recent data stuff, because now if you go back to genetics, it's actually very tight. And then we still see the chromosome 6 and 8 and 10 QTLs. And then now if you see the Manhattan, we see the chromosome 6 QTL and chromosome 10 QTL, and 8 goes away. And if you look at do some research, we found out that chromosome 8 QTL was temperature related, that sensitivity on that, it's not that photoperiod related. So it's actually the 6 and 10 is the common QTL that behind all this change of the flowering time. And then now this actually, I don't know if you get it, this is the most interesting slide we have. It just captures a lot of information here. Again, there's Puerto Rico, Kansas, Iowa. And then here, so remember we have 2 major QTL control this photoperiod response, right? And then chromosome 6, it is T allele with TX430, or P allele, which is P89, 8012. And there's something you cannot see here, but we'll just wait here. And then on the Kansas, you can see that if the T allele coupled with the T allele, and then this is a P allele coupled with the P allele, and then this another one is in Iowa, they have a crossover interaction. But if I enlarge this part, you see actually there's actually going downtrend. This actually, the red line is here going up, and this one red line going up here, and this red line going up here. So what we're saying that there's a QTL by QTL interaction. This epistasis together interact with the environment, give you this G by E interaction and the parents. Okay, and then so we went to the literature, and then Jianming's group published this paper on NE1, which is a PRR37 gene, and then we think that this is a chromosome 6 QTL we detected too, and this possibly to be the chromosome 10 QTL, that this candidate gene underlying this one. And then if we grew this TT allele together, this is actually the TX430, TT allele, PP allele, TT, and TP and PP. So this is just different QTL combinations. You grew them in different place. They'll show you different flowering time response. So now we try to work with some of the crop modelers, physiologists. So they're doing this episode modeling. We try to combine that information into the modeling of the G by E interaction here. The last one will be quick, but it's actually interesting. Why we talk about this one is the bioenergy genomics. This was a funded project by the USDA DOE feedstock genomics program. I think the bioenergy crops, they're relatively new. They're not like this, regular sorghums or the crops that would be green sorghum, corn, maize, rice, wheat. They're new. So how do we actually tap into this vast plant germplasm collections for the biomass crop improvement? They provide the opportunity for us to do something slightly different, because breeding is still breeding, it's still growing, it's great, right? We can do a little bit different. So how do we increase the information content or information contained in the data so that we can extract maximum information or knowledge, help us understand the germplasm, and then establish the genotype-phenotype relationship? This sounds like lofty, but what we did is actually You know, in the green database of USDA, there's a lot of accessions. So we go in, we select first 1,000 accessions, and I'll explain that later on how we selected 1,000 accessions. And then we did GBS on 1,000 accessions. And then we did something different, is a selective phenotyping. You're probably familiar with selective genotyping. That's when 20 years ago, we did not have enough money to genotype every individual. So we say, we'll genotype these trait extremes. But this is different, okay? This is actually the genotype a larger accession first, and then select a subset to do phenotyping. And then we can do this field-based study and also do the NRI or high throughput phenomics on this, this prediction of this biomass compositions. And then you can do GWAS and then haplotype enrichment. That's the scheme of the project. So the reason how we narrow down this germplasm is actually just to start right. We're working with the germplasm bank specialist, and then they have a lot of information. So we have 44K accessions of sorghum. About 78% of them are photoperiod sensitive. That means they have a potential to be used for biomass production. Grow them in temperate regions. So we narrow this 34K down to about 33,000 accessions based on their flowering time, photoperiod sensitivity, seed availability, and germplasm information. Later on, we narrow it down to 1,000 of them where we can get enough seeds for our, do the phenotype and genotype. That's a, going smaller process. And then what we did selective phenotyping is actually we did trait evaluation only on individuals that contain the maximum genetic information, which is 300 of them. So genotype large population, select subset. This selective phenotyping actually in the literature, you talk about under the biparental population or the large diverse population, you want to maximum the allele numbers, gene diversities, or recombination breakpoints, right? And then there will be increase of mapping power and resolution compared with random selected 300. So you have some design thinking going into the process, right? And then also we would expect it to increase your prediction accuracy. That's 1,000 to 300, your phenotype is 300, and then you can do predictions for the rest of the 700. So if you look at this, because we have all this data, you can say this genetic relationship, you calculate the kinship and the two-way cluster. This will be what you look like of the close to 1,000 accessions. There's some relatedness accessions, but then if you do this 300 data set, they're pretty clean. There's less relationship building in within the set. So this is a set that we want to, you can see that this is cluster, this stems, and all this hierarchical structure here. They're so different. So you want to actually focus on phenotyping on this, select the subset. And again, this slide, I don't want to bother with those details. What we did is actually, with 300, we can actually calculate the prediction accuracy just by manipulation of data called cross-validation. This is fairly simple. And you can see there's a different methods for this cross-validation. These are pretty much the same thing. You can see the stock numbers because we have 2-year data and high heritability or repeatability of this data. We have prediction accuracies very high. But then it's raw biomass on 1 year, we really have problems. So we actually have the prediction accuracy 0.4. And then again, depending on you're optimistic or pessimistic, this 0.4 should never be compared to 1, okay? The prediction accuracy should never be compared to 1. Everything is predictable and we have nothing else to do. It should be compared to 0. Again, this is something that we have to get the message across. Every time people think the prediction accuracy is low, they're not happy, but I'm happy because either you have a prediction or you have non-prediction, okay? 0.4 is good enough, actually. It's not good enough, it's good, it's always good, better than zeros. So here is the scheme here. So remember, we were going down from large accessions to the smaller numbers. Now, once you establish a robust 300 prediction models, you can go back to predict the rest of 700. And then if you do some empirical validation with this 700, you check whether you can still go further. And you retrain your data set. You can have another updated training, the prediction model. You can expand to the 3,000. Maybe, that's just a theory, right? To get this done, you require a lot of field work and the computational work. But again, how we did this genomic assisted approach to the genetic improvement of biology crops is germplasm information, selective phenotyping, GBS, genomic prediction, and also at the same time, you can do GWAS if you want, because you can actually, you have genotype, phenotype of the 300, right? This is probably last slide before my acknowledgement. So I think quantitative genetics has a significant role in the area of high throughput genotyping and phenotyping. Some of the things I haven't touched because we haven't really think of good ways how to handle this one. But I give examples of epistasis, GYE, and the rare variants, and also the genomic selection for the germplasm enhancement. And then action, again, this is the plant breeding symposium. I always, try to treat myself as one of the graduate students. So you get younger, right? And then take courses in quantitative genetics and related areas, plant breeding, and all this. I get a checklist. I think many of the attendants have taken that one. Study one or more of those textbooks I listed in the first few slides, those textbooks. It's always amazing that before I give a lecture, I'm sure that it's all in the same thing. Before we actually go teach students, we go back and read all those book chapters, make sure we understand again, refresh our memory, and before we discuss. And then learn how to answer a question in genetics and breeding without molecular marker or QTL. This actually is very interesting. I mean, every time we have a question, interview and stuff, I mean, we can answer questions without molecular marker. Even I showed you a lot of genomics, cloning, gene structure change, protein stuff. But you can answer questions without having to refer to molecular marker or QTL because there's genetics. There's old papers that are there that are just great, right? You have to learn. And also get used to draw a flow chart and derive equations if you want to be good on quantitative genetics, if you want to be very competent in handling this big data, this sequencing data and the high throughput phenotyping data. And also it's helpful to know how to program. I was used to program C++. I know that now these days you have to play with R. Yesterday we had a dinner and students were saying how difficult to learn R and then, but you have to update your skills all the time. And then, so read papers and discuss with others, attending meetings, always helpful. Here I listed some of the equations actually. I mean, they're textbook equations, right? And there's this quarter, half quarter, and then this additive variance, dominant variance. This would be the marker class mean contrast of the QTL mapping of the F2s. This is the linkage disequilibrium calculations, the R-squared calculations. I mean, they seem to be just so distant from us. But they actually, if you read some of our papers, you see this actually exactly things that are still very relevant with the genomic data, with all this high super phenotype data. It's very relevant. We should actually train students not afraid of the using equations to establish some level of expectations, then use the improved data to test whether this expectation are correct or not. Okay. With this, I'd like to give acknowledgement. Over the years, we've been funded by the NSF Plant Genome Program, led by the Mike Scallan Shoot Apical Meristem project. These are the co-PIs. And then when I was in K-State, we had a lot of collaborations with folks there at K-State, the breeders, and then the crop engineering specialist, and then the crop production physiologist, and then Frank White, and then this Scott Bean, Mitch now moved to Purdue, and then Howard Trick. Sorry, this is actually the, why I get this wrong? Nebraska. Oh, no, KSU, actually, KSU for Howard Trick did the transformation of adopters for the gene validation. And then Jeff Morris now is the position at Sorghum Genetics position. Mingli Wang and then Tom Clemente here from Nebraska. These are the earlier collaborators and Rex and others. This is the human geneticist. We were working on this human charge consortium data. And then these are the students and these are current grad students, Xin Li and Matt. We were funded by different agencies. Thank you very much.
Okay, we have time for a couple questions. There's any questions?
In your last trapezoid, you show the frequency of A and little a.
Uh-huh.
But
Yes.
when you and that would be true for QTL, biparental QTL
Yes.
mapping.
Uh-huh. Mm-hmm.
And then you point out in your earlier discussion, you have a rare allele with a large effect in GWAS.
Yes.
That approach.
Yeah.
There's a conundrum about that because you're analyzing accessions. So if you have a rare allele with a large effect and you walk out in the field and look at the individuals, the 1,000 or so, and you have one individual that's 1 out of 1,000 that shows a strong height or whatever, can't you just find that rare allele with a large effect by inspection rather than quantitative genetics?
Oh, here.
Your slide of the rare allele distribution.
Yeah. So here actually, this is the 50-50 would be the one, textbook examples, but actually the more relevant one is here you have a PQ that involved. And this PQ in the bioinformatics populations, we have half-half, but in the GWAS it will be difficult.
Go back to your slide where you have the allele effect size and the
number one.
Right. Oh, too far. You were there, right there.
This one?
Next one. Where your theory.
Okay.
Rare alleles. You had the curves.
Oh, curve. Okay. Here.
There, right there. Okay. Now you show a very low frequency variance, common variance, and then the rarer variance. And what you're looking for is the large effect one, assuming it's a positive one, right?
Yes.
Because those rarer variants are what breeders would use instead of the common ones to get to the phenotype faster. So when you're going into the collection, you're looking for rare alleles, basically. And if you get lucky, you hit them in your first QTL mapping population. So, you're saying you can explain the dark matter of the quantitative genetics universe by the route that you did, but from a practical perspective, we want to find large, non-deleterious effect alleles in the germplasm for immediate use in breeding.
Yeah.
Right? Would you agree with that as an assumption?
Yes.
And so, one of the problems with GWAS is those alleles in paper in silico, it's hard to find those, right, without actually looking at the lines in the field. So it involves not just phenotypic variation, but actually going out there and seeing the particular lines like you did. So I just wanted to make that point because sometimes you can get swamped with all the mathematical considerations without looking at really the field itself.
Yes.
I mean, there's computer breeders and then there are field breeders. So
I'll take that as a comment. So I have not, I was just saying that this rare variance, although it has a large effect, it's not necessarily show up in your field. It might be masked by other allele, other effect. This is something that here we can show that rare variance has a large effect, but whatever can show in the field is the mutant, that severe alleles that will show up right away. So this is slightly different, but I take that comment. Yeah, thank you. Yeah.
All right, one more question.
Maybe I didn't quite understand what you did, but I think when you're partitioning the variation in your human data set among common variants and low variants and so forth, I mean, you're just calculating the kinship matrix, right, with the different types of SNPs that fall into the different categories?
Yes.
So, I mean, I guess the question is the contribution of rare variants to total variation and their importance relies on the fact, or assumes that maybe they have large effect, but when you calculate the kinship matrix, you're shrinking their effect back, so you're constraining their effect. Right?
Okay. So there are always two levels of effect size. When here we talk about the genetic effect size, it's almost think about the small a, big A to small a, and divided by 2. That's effect. But then within this calculation of the contributions, we're talking about actually was weighted by the allele frequency already. You contribute to the genetic variance. It's a 2pq alpha square. It's actually already being weighted by the frequency. Is that clear? I'm not sure if I answered the question.
But you partition this variation by calculating several different kinship matrices, right? So you're not allowing so the rare variants are not allowing them to have their full effect that they might have if you had treated them as a fixed effect.
Okay, great. But yeah, that's great because remember your total phenotypic variance was also is the whole population. Even that the rare variants might have a potential large effect under equal frequency. But because their frequency is low, and because that the evolutionary theory, where the deleterious alleles or where alleles they tend to negative alleles to be selected against, so that they don't have a high frequency. So this weighting by the frequency to calculate its contribution is relevant to that specific population. That's what we get. Yeah. Thank you.
All right, thank you. Let's give Dr. Yu another round of applause.